The overall goal of this procedure is to provide a simple Y-maze assay to test olfaction in Drosophila melanogaster. This is accomplished by first starving flies for 16 to 18 hours so they are motivated to explore new environments with volatile stimuli. The second step of the procedure is to assemble the Y-maze setup containing the odor sources on filter paper, such as acetic acid in solvent versus solvent alone. The third step is to quickly and gently introduce the flies into the loading vial. After several hours, the final step is to score the fly's location in the maze. Ultimately, the results can show a response toward odorants from the data obtained with the Y-maze.
We first had the idea for this method when we decided to have a simple and inexpensive method to test the olfactory response of Drosophila. An advantage of this technique over other options is that it allows the flies to make a decision without too much stress.
Demonstrating the procedure will be Mégane Simonnet, a PhD student from my laboratory.
For this protocol, have ready an isogenized fly line with stable, robust behavioral phenotypes. Any such stock will serve as a reference control for the olfactory test. For each genetic strain to be used for the experiment, back-cross the strain to the reference strain for 5 generations. Pair single experimental strain females to a few isogenic males for each back cross.